In a world where species are disappearing at an unprecedented rate, scientists have developed an extraordinary conservation strategy hidden from public view. Deep within specialized laboratories across the globe, vast collections of cells from thousands of endangered animals rest in suspended animation at temperatures of -196 degrees Celsius. These frozen zoos represent one of conservation's most forward-thinking approaches, banking genetic material today to potentially rebuild what humanity has lost tomorrow. Join us as we explore this remarkable intersection of cutting-edge technology and conservation, where cells preserved in liquid nitrogen may hold the key to saving species on the brink of extinction. Our story begins in 1972, when most conservation efforts focused exclusively on protecting habitats and breeding endangered animals in captivity. Biologist Kurt Benirschke at the San Diego Zoo pioneered a radically different approach. He began collecting and freezing cells from rare and endangered animals, creating what would become the world's first comprehensive, frozen zoo. Benirschke's vision was remarkably prescient: preserve the genetic material of vanishing species before they disappeared completely, banking biological information that might someday prove invaluable. While his colleagues viewed the project with skepticism, Benirschke persisted with extraordinary foresight, often stating, "We're doing this for people who aren't here yet, using technology that doesn't exist yet." This visionary scientist understood that even if we couldn't immediately use these preserved cells, future technological breakthroughs might transform them into powerful conservation tools. The process of preserving cells for a frozen zoo involves several sophisticated steps. First, researchers take small tissue samples, typically skin biopsies, from living animals. These samples are then processed to isolate fibroblast cells, connective tissue cells that are relatively easy to culture and preserve. The isolated cells are cultured in laboratory conditions until they multiply sufficiently. Next comes the critical step: suspending these cells in a cryoprotectant solution designed to prevent the formation of ice crystals that would otherwise destroy cell structures. Finally, the protected cells are stored in liquid nitrogen at the extreme temperature of -196 degrees Celsius, where all biological activity essentially stops, allowing the cells to remain viable for decades or potentially centuries. The technical challenges were substantial in the early days. Early cryopreservation techniques frequently damaged cells during freezing and thawing. Researchers had to develop specialized media that could protect cellular structures while maintaining viability after thawing. The development of programmable freezers that could control precise cooling rates, approximately 1 degree Celsius per minute, proved critical for successful preservation across diverse cell types from different species. What began as a genetic archive has evolved into a powerful conservation instrument with multiple applications. The San Diego Frozen Zoo now houses more than 10,000 cell cultures, representing nearly 1,000 species and subspecies. These include cells from the now extinct woolly bird from Hawaii and the northern white rhinoceros, which has dwindled to only 2 living individuals, both female and unable to reproduce naturally. The frozen collections have transformed from academic curiosities into vital resources as biotechnology has advanced. In 2020, researchers achieved a significant breakthrough when they created embryos of the northern white rhinoceros using frozen sperm and eggs harvested from the last living females. This technology, combining assisted reproductive techniques with cryopreserved materials, represents one of the most promising pathways for reviving functionally extinct species. As the original vision of Benirschke materializes, these frozen repositories demonstrate their value not just as archives of what has been lost, but as active tools in the fight against extinction. Perhaps most surprising is the significant contribution of frozen zoo materials to basic science and research beyond direct conservation efforts. In 2013, cells from the frozen zoo were used to sequence the genome of the amakihi, a Hawaiian honeycreeper bird. This research revealed how these birds evolved resistance to avian malaria, information now guiding conservation strategies for related bird species still vulnerable to the disease. The applications extend far beyond conservation. Researchers have utilized frozen zoo cells to investigate evolutionary relationships between species, study chromosomal abnormalities, and even explore comparative oncology, examining why particular species appear resistant to cancers that plague humans. For example, studies of elephant cells from frozen collections have revealed multiple copies of the p53 tumor suppressor gene, potentially explaining why these large, long-lived mammals rarely develop cancer despite having many more cells than humans. These discoveries demonstrate how preserved biological materials can yield insights that benefit both wildlife conservation and human medicine. Perhaps the most technologically ambitious application of frozen zoo materials involves induced pluripotent stem cells, or iPSCs. Scientists have successfully reprogrammed frozen fibroblasts from several endangered species into stem cells that can theoretically develop into any tissue type. In 2011, researchers created iPSCs from frozen cells of the northern white rhino, potentially allowing for the creation of reproductive cells, sperm and eggs, from skin samples. This represents a revolutionary capability for species with few or no reproductive individuals remaining. The implications are profound. Theoretically, scientists could create diverse offspring from a wide genetic pool using only preserved skin cells, even if the original animals are long deceased. This technology, combined with surrogate pregnancy in related species, offers a pathway to restore genetic diversity in highly endangered populations or potentially revive extinct species. While still experimental, these techniques demonstrate how rapidly advancing biotechnology is transforming frozen cell collections from passive archives into active tools for species restoration. The frozen zoo concept has spread globally, with major repositories now established in the UK, Australia, Japan, and several other countries. The Frozen Ark Project, founded in 2004 at the University of Nottingham, coordinates many of these efforts, creating standardized protocols for sample collection and preservation. This international cooperation ensures that genetic material is preserved according to best practices regardless of where it's collected. One lesser-known but equally important facility is Brazil's DNA Bank of Wildlife of Pantanal, which focuses specifically on preserving genetic material from the unique fauna of the world's largest tropical wetland. This specialized approach highlights how biobanking has evolved to address regional conservation priorities, protecting the genetic heritage of specific ecosystems. Unlike traditional museum specimen collections, these biobanks require continuous maintenance and energy input. The San Diego Frozen Zoo maintains backup generators and duplicate samples at different locations to protect against catastrophic failure. The annual operating cost for maintaining such collections typically exceeds $500,000, raising questions about long-term sustainability and funding security. The coordination between these facilities presents unique challenges that go beyond the science of preservation itself. The CryoArk's Biobank network in the UK has pioneered an integrated database system that tracks not only the existence of samples but also their quality, associated data, and availability for research. This infrastructure development, while less glamorous than the creation of northern white rhino embryos, may ultimately prove equally crucial for the long-term utility of these collections. Proper data management ensures that researchers decades from now will know exactly what genetic material is available and how to access it. Some facilities have specialized in preserving genetic material from domesticated animals as well. The Svalbard Global Seed Vault has an animal counterpart in the form of the Animal Genetic Resources Cryobank, which preserves genetic diversity from livestock breeds facing extinction due to agricultural standardization. This recognition that domesticated biodiversity also requires conservation represents an essential expansion of the frozen zoo concept, acknowledging that traditional agricultural varieties and breeds contain valuable genetic traits that might be needed in the future. The concept of a frozen zoo raises profound ethical questions that researchers and conservationists continue to grapple with. Critics argue that these repositories could divert attention and resources from more pressing conservation needs, such as habitat protection and addressing climate change. There's also concern that the existence of genetic backups might reduce the perceived urgency of saving species in their natural habitats. After all, why protect an endangered species in the wild if we have its DNA safely stored away? Other ethical considerations involve the potential revival of extinct species. The technology to create northern white rhino embryos exists, but successfully implanting them in surrogate southern white rhinos remains challenging. If scientists succeed, would the resulting animals truly represent the species, given that they would grow up without the cultural knowledge usually passed from parents to offspring? Would they be merely genetic replicas lacking the behavioral and social aspects that define a species? The question of prioritization also looms large. With limited resources, which species deserve a place in these frozen arks? Most collections have focused on charismatic vertebrates like mammals and birds, while invertebrates, which constitute the vast majority of animal biodiversity, remain underrepresented. Recognizing the limitations of focusing primarily on mammals and birds, frozen zoos have begun expanding their collections to include a wider range of species. The San Diego Frozen Zoo has recently added coral cells to its repository, acknowledging that these colonial marine invertebrates face existential threats from climate change and ocean acidification. This expansion represents an important shift in perspective, recognizing that ecosystem health depends on preserving biodiversity across all taxonomic groups. Recent innovations include the preservation of gut microbiomes from endangered species, recognizing that an animal's associated microorganisms play crucial roles in digestion, immunity, and overall health. The American Museum of Natural History now maintains a frozen collection of amphibian skin microbiomes, which could help develop treatments for the deadly chytrid fungus decimating frog populations worldwide. These developments highlight how the frozen zoo concept continues to evolve, moving beyond preserving just the animal itself to capturing the complex biological systems that support it. As our understanding of biodiversity deepens, the scope of what needs preservation expands accordingly. As extinction rates accelerate due to human activity, these frozen archives of genetic diversity represent both a safety net and a promise that even if we fail to save species today, we might preserve enough of their biological essence to give them a future tomorrow. Emerging technologies continue to expand the capabilities of frozen zoos. Advanced genome editing techniques like CRISPR may someday allow scientists to correct genetic defects in endangered species with small population sizes and high levels of inbreeding. Artificial wombs, currently being developed for human medical applications, could potentially provide new pathways for reproducing endangered species without surrogate mothers from related species. The integration of artificial intelligence into biobanking is improving sample management and helping predict which species are most at risk of extinction, allowing for proactive collection before populations collapse. Perhaps most importantly, frozen zoos are increasingly seen not as alternatives to traditional conservation but as complementary approaches. The ideal scenario involves preserving species in their natural habitats while maintaining genetic backups as insurance against catastrophic events. The frozen zoo stands as a testament to human foresight and technological ingenuity, but also as a sobering reminder of our species' devastating impact on the planet's biodiversity. The frozen zoo represents one of humanity's most innovative responses to the biodiversity crisis, a technological arc carrying the genetic essence of thousands of species through turbulent times. From Kurt Benirschke's visionary beginning in 1972 to today's sophisticated global network of biobanks, these collections have evolved from simple archives into active conservation tools with applications ranging from reproductive technology to medical research. As we've explored, the science behind cryopreservation has advanced dramatically, enabling breakthroughs like the creation of northern white rhino embryos and the reprogramming of skin cells into versatile stem cells. Yet these technological marvels also raise profound ethical questions about how we value biodiversity and allocate limited conservation resources. The frozen zoo concept reminds us of both human ingenuity and our responsibility for the extinction crisis. These facilities represent neither a perfect solution nor a substitute for protecting wild habitats, but rather a complementary approach in our conservation toolkit. As climate change and habitat destruction continue to threaten countless species, these frozen repositories stand as both a promise and a warning that while we may preserve the genetic blueprints of biodiversity, the rich tapestry of behaviors, relationships, and ecological functions that define living ecosystems requires our immediate protection. The future of conservation will likely involve both traditional approaches and these high-tech arcs, working in concert to preserve the extraordinary diversity of life on Earth. Thanks for watching. Please like, subscribe, and hit the bell icon for updates. Your feedback is appreciated. We hope to see you again.